So good morning, everyone. All right, I'm glad to see everybody here in the room. I know that there are individuals that are on Zoom. I just want to say thanks for coming, and hopefully everybody will benefit from today's meeting. Uh, First, I want to welcome you to the 21st annual public meeting of the long-term plan for achieving water quality goals for the Everglades protection areas. I'm Robert Shuford, lead scientist here at the district. And this meeting is held for you, the stakeholders, to inform you of the progress that we're making in improving water quality into the Everglades protection area. All right, before I begin, everybody in here probably already knows, but I'll just, reiterate and orient you a little bit. Through these doors to the left, we have restrooms, vending machines, water fountains, and a cafeteria. So if I get too boring, just grab a coffee or something. The only thing I ask is that you don't bring food in. We don't necessarily want to have a lot of eating going on. So the agenda for today, I think you all have an agenda, but we'll have a total of 7 presentations today. Those presentations will range from the system itself to the detailed work that's being done in the features that that are structured for improving water quality. In particular, if there's areas of interest that you have, we ask that you pay attention to the contact information, which is a slide. The very last slide of every presentation will be a contact email for the presenter. And so if you actually have an area of particular interest or need additional information, we ask that you'll contact the presenter to follow up. Okay. Our first presentation today is going to be me, and I'm going to share with you systems conditions that sort of laid the foundation for the work that has been done this past year in the system. So the discussions today will deal with the long-term plan, which was actually enacted in the 2003 Everglades Forever Act, EFA. In 2012, they expanded the language of the long-term plan and included restoration strategies. And restoration strategies in and of itself included 13 projects that will be completed over a 13-year time frame. So the results of all of this work, we would ultimately end up with 62,000 acres of STAs, stormwater treatment areas, and about 116,000 acre feet of flow equalization basin storage, FEBs. In addition to those projects, we've included some work above and beyond restoration strategies. We've called that refurbishments, but that's work that we've identified and enacted on to help bolster and continue this water quality capabilities. Excuse me. In this slide, you'll see the area, an area of concentration. Most of you guys know this tan area that we call the Everglades agricultural area. And it's within this area that most of Restoration Strategies projects lie. You'll notice that Everglades EAA is bounded to the north by Lake Okeechobee and to the east and south by the conservation areas, water conservation areas 1, 2, and 3. So we'll be talking about that information in terms of looking at the system itself. All right, so, well, you'll hear a lot about water year. And so just to make sure everybody's on the same page, we look at a water year that ranges from May to April of the subsequent year. This is a depiction of water year 2024. It starts May 1st of 2023 and ends April 30 of 2024. Within that water year, we have what we call wet seasons. We have a wet season and a dry season, and those seasons are partitioned as May through October. In general, May through October is our wet season, and November through April is our dry season, right? All right, so as I mentioned previously, the EAA is bordered to the north by Lake Okeechobee and to the south and east by the conservation area. So I just want to give you an overview of what the schedule looks like. So each of those features have their own schedule. And in general, we try to, I'll just give you an orientation of this slide. Along the side here, we have elevations, the y-axis elevations, and then we have these multiple colored bands that sort of dictates where the lake is at any point in time. So we have this red band, which is the high lake management band. Band below that is what we call the high band. Then we get into an intermediate band, and this larger dotted band is the low band. We get into something called base flow, which is this solid line. And then finally, the lowest band that we try to operate within would be called the beneficial use band. Over this past year, you'll see that we've largely operated, well, let me just add that typically we try to, as we're approaching a dry season, we try to make sure we have elevated levels, and the stages are generally lowest as we approach the wet season in anticipation of rains to restore the system. This past year, we've actually operated pretty much between the intermediate band and the low sub-band. And of course, in previous years, we've actually had a much broader swing. For instance, 2022, we actually ranged between the intermediate band and as low as the beneficial use band. Similarly, when you look into the conservation areas, they have their own schedule. And again, they have their zones, zone A, zone A2, and then this bottom band which is the water storage floor. And those, again, they follow a similar pattern. They have the highest stages, or attempt to have the highest stages as we approach the dry season, and then it lowers as we approach the wet seasons. For some general information, the green traces in these slides represent canals, and the blue traces represent the stage that's actually in the marshes. Stage, runoff, they're all impacted by rainfall, and this graphic shows the rainfall that we've received during the dry season of water year 23, as well as pretty much to date the water year 2024 conditions. And you can see here wet season, prior to the start of this water year seems to be starting early. The wet season in general was pretty normal, and our dry season, largely as a function of El Nino, has actually been started started normal, and then, of course, we've had several, late season storms or early dry season storms that has actually already influenced our stages in the system. Back in early November, we had what they call a wake low urban flooding event. And of course, December, there was the high rains. And you guys probably remember we had flooding in the airport in Broward. So those events kind of marked the beginning of our wet season this year, and we've had several events thereafter as we moved into the year 2024. Okay, this slide depicts the dry season rainfall in water year 2023. Again, if you remember, the dry season pretty much runs from October of the previous year into April of the subsequent year. So the graphics start as early as November of 2022. Overall, the dry season, we experienced our greatest rains coastal during the water year 23. This little white box that you see here sort of demarcates our area of interest. Rainfall in those areas, for the most part, has been normal during the dry season for water year 23. November started with a bang, and April ended with a bang for that dry period. So those were our wettest months, followed by the extremely dry conditions that persisted from January through March. Wet season or wet season rainfall is depicted here for water year 2024. And you'll see again, this red box demarcates our area of interest, coupled with the red boxes that are in each slide or each graph. We started May through August with very normal conditions for the wet season, and then kind of progressed into drier conditions as September and October rolled in. And this is a relatively normal pattern for our wet season. In general, the EAA kind of experienced rains between the 40 to 50 inch range range. And as I said, the drying began in September and October, and that year we ended with a 94% of normal rain conditions. And then dry season, which is the current status or the where we are now of 2024. Like I said earlier, it began with a bang, and we can see that in November of this year, we had elevated rains that have literally persisted through to the end of this. These graphics go to the end of January, but we've had sufficient rains. By mid-January, we had about 168% of normal in terms of rains, which gave us a 4-inch surplus. And like I mentioned, the greatest rains kind of kicked this season off in November. All right, so all these, I show this rain information because, like I said, rain influence runoff and what these STAs and FEBs are exposed to. And so I just want to show in these last couple of slides, kind of summarize the rainfall and the inflows during the past 3 water years. In this graphic, at the top graph, the y-axis is the total rainfall. The total inflows. I'm sorry, into the STAs, into the features, and it's measured in basically thousand per acre feet or thousands of acre feet. So, for example, in water year 2022, we had basically 885,000 acre feet of water that it happened in the wet season being the blue, dry season being the yellow. The green, in the lower graph, we have the rainfall in inches, the green representing the wet season conditions, and the orange representing the rainfall in the dry season. And as you can see here, for the most part, our wet season water years, the rainfall has been relatively normal, and for the most part, our wet season runoff or wet season inflows have been, relatively stable. We got a bit more in 2024. It might be associated with El Nino conditions, but I won't go into details on that. The beauty is we'll learn more about how these changes actually impacts the system or the systems in the next presentation. But again, similar conditions in water year 22 and 23 in terms of the wet season. Well, really in terms of wet season and dry season, but we've got some increase, a major increase in water year 24. Okay, and as I said previously, up through January, mid-January, we already had for water year 24, about 168% of normal in terms of rainfall. And then finally, we have the water year, and just looking at general inflows, based on runoff, as well as lake water releases. Lake water releases speaks to our ability to move water to the south. And again, in this depiction, the runoff is demarcated in blue, and yellow represents lake water. Now keep in mind, runoff is influenced by a number of inputs, but it's not limited. It's influenced by a lot of things, but it's not limited just to rain. And in this case, we see the runoff is the dominating the inputs for 2022, 23, and 24, considering our rains, in general, were normal. 2022, we literally had 90% of normal for the year. 2023, 103 for the year. And then, of course, in 2024, even though we haven't completed our dry season, we have a few months left. Overall, for the year, we're at right at about 168% of normal. So these are the conditions that will influence the information, the data that you see in subsequent presentations. And you'll get more in depth into kind of drivers and those things. But I just wanted to give you an overview of the system itself. So our next presentation is going to take a greater look into these runoff and influence impacts. And that presentation, that presentation will be done by Mr. Jake Dombrowski. He's a senior scientist in applied science, and he is your next presenter. So I'll give the floor to you. Okay.
All right. Morning, everyone. As Robert mentioned, I'm Jake Dombrowski. I'm a senior scientist in the Applied Sciences Bureau. Today, I will be giving the update on the Everglades stormwater treatment area's performance. So starting out and talking about STA performance, I'm primarily going to be sharing flow volumes, TP loads, and flow weighted mean concentrations. So this is going to be for each of the individual STAs. You can see them on the map here in the olive green color, going from STA 1 East near Wellington out to STA 5-6 in Hendry County. So I might refer back to just the concentration at a few points, but I'm always going to be referring to that flow weighted mean throughout the presentation. So for these parameters, we'll look at yearly and monthly variation. This is going to focus a lot on the district's water year. As Robert mentioned, that runs from May 1st of the previous year up until April 30th of the current year. And finally, I'll share some of the construction and other operational restrictions that impacted the STAs. So just to orient you a little bit in this first slide here, a lot of these slides are going to look very similar. So the chart at the top is the concentrations with the inflow concentrations in red, outflow concentrations in blue. I'm going to be sharing data from water year 22 through the current year, water year 24. So water year 24 is still a partial year. In this presentation, data will be included through December 31st of 23, and that will have provisional or preliminary data. The table at the bottom here shows a few more of these parameters, such as the inflow volumes, the TP loads, and then the percent reduction in concentration and load by the STA. So for STA 1 East, looking at water year 24, so far, water year 24, we've seen higher inflow volumes compared to last year, but in general, 1 East has been receiving lower flows in recent years compared to its period of record. I'm going to touch a bit more on that in a minute here. In terms of concentrations, we're actually seeing lower inflow concentrations coming into STA 1 East, and then lower outflow concentrations as well relative to recent years, while the percent reduction in TP concentration and load is fairly similar to previous years. So this slide is going to highlight the monthly variation. There's 3 different plots here with the concentrations at the top, the TP loads in the center, and that's in metric tons, and then flow volumes at the bottom in thousands of acre feet. The inflows are still in red and outflows are still in blue. So as I mentioned on the previous slide, STA 1 East hasn't received quite as much water in recent years. This is due to a lot of ongoing construction within the STA. A lot of that construction's wrapping up, but we're still reestablishing some of that vegetation, so there's still some operational restrictions there. And at the same time, STA 1 West, directly next door, has had a lot more operational capability, and they both serve the same basin, so more of those flows have been going to 1 West instead of 1 East. And the one thing I did want to highlight on this slide here is that blue line at the top here, that's the outflow concentration for STA 1 East. And so far in water year 24, it's very low and very stable. Looking at operational restrictions, so these are going to be for the calendar year 23, not the water year. In STA 1 East, the western flowway is still offline. This was for some earthwork that was done, and that earthwork's complete, but as I mentioned, we're still reestablishing some of that vegetation in that flowway. In addition to that, we had periodic restrictions in the eastern and central flowways. This was primarily for vegetation management, as well as a restoration strategy science plan study, and then nesting by some of our migratory birds. And there's an example on the bottom right here of a black-necked stilt and her chicks. And then the photo on the bottom left highlights some of that vegetation establishing in cell 7 of the western flowway. Moving on to STA 1 West. So you can see in water year 23, water year 24, much higher inflow volumes compared to previous years for STA 1 West. And looking at the concentrations, the inflow concentrations actually substantially lower compared to recent years, and then we're seeing a lower outflow concentration as well in STA 1 West. And then with those lower inflow concentrations, we do see a lower percent reduction for this STA. And looking at the monthly trends, so as I mentioned, higher inflow volumes so far in water year 24, and then water year 23 as well. So with all that ongoing construction in STA1 East, it's limiting the flows that can go in that direction, so it's been going to STA1 West instead. And within 1 West, we've had a lot of refurbishments projects that have recently been completed, increasing our operational capacity there. And wanted to highlight on this slide as well, the blue line at the top here for STA1 West. You can see a nice clear declining trend over the past couple of years, but especially in water year 24. In terms of operational restrictions, all flowways were online, but there were periodic restrictions in all flowways. This is primarily related to a few refurbishments projects that impacted the northern, western, and eastern flowways. And while those are complete, we still have a lot of planting going on there. In addition, we had some extra planting occurring in the northern flowway. So the photo on the bottom left here highlights some of those EAV strips going in the inflow area. And then we've had construction related to the expansion number 2 of STA1 West, which has impacted operations for cell 8. And I've included a shot here of the new pump station, G780, that's going to serve that expansion number 2. And finally, we had some blackneck stilt nesting throughout STA1 West. All right, moving on to STA2. So STA2's had a pretty wet year so far. Not quite as high as last year, but with the El Nino pattern that we're in, it's likely that those flow volumes are going to be quite a bit higher as water year 24 closes out. The inflow concentrations are quite a bit lower for STA2, while the outflow concentration is comparable to water year 22, but lower, definitely lower than last year. And the percent reductions follow a pretty similar trend, with them being higher compared to last year. But one thing I want to highlight with STA2 and STA3-4 in a few minutes here is the benefit of the A1FEB. So the A1FEB helps to capture a lot of those peak flows in the wet season and reduces some of that concentration and load coming into the STA. So you can see in general, the inflow concentration, the red line up here, it's typically much more stable for STAs 2 and 3-4 compared to other years. And then the outflow concentration STA2, the blue line at the top, has been quite a bit lower and stable in STA2 this year. In terms of operational restrictions, Flowway 2 is offline for an earthwork project, so that earthwork has been completed, but restrictions remain for the vegetation being reestablished there. And there's a photo on the bottom left here showing some of those plantings from earlier this year for the region that had the earthwork done in the inflow area. And so in addition to that, we had periodic restrictions in flowways 1, 3, and 4. This was primarily vegetation management activities, and then we also had some canal plug refurbishments in flowways 2 and 3. And there's an example photo in the center here with some of the recently refurbished plugs. And I always like to include a photo of our tropical spring breakers that make a pit stop in STA2 every year. All right, STA 3-4, so very wet year for STA 3-4 so far. The inflow volumes are substantially higher compared to previous years, and that number is only going up. At the same time, we are receiving lower inflow concentrations compared to previous years, and then the outflow concentration's been trending downwards, although it's usually quite low for STA 3-4 as it is. And then the percent reduction with those lower inflow concentrations, the percent reduction is a little bit lower this year. Looking at the monthly variability, again, want to highlight the benefit of the A1FEB. Those inflow concentrations are generally on the low side, but very stable coming into STA 3-4 thanks to the A1. In water year 24, we've had some pretty high flow volumes that continued throughout the summer, running into the fall, and then we're still seeing those higher flows occurring, being in the El Nino pattern. And last thing to highlight for 3-4 is that outflow concentration, very low and very stable in water year 24. In terms of operational restrictions, there's not too much to say. The eastern flowway has been offline. This is for a vegetation rehabilitation project through a managed drawdown. So that drawdown's now complete, and we're just in the final stages of getting the vegetation ready for normal flow through. The bottom right photo here shows some of those plantings in cell 1A in the inflow area. And then the other photo here is one of a drone shot of the mixed marsh in cell 2B. All right, our last STA, STA 5.6, and the one thing that always jumps out when looking at STA 5.6 is how big those red bars are. So inflow concentrations coming into 5.6 are substantially higher than the other STAs. In terms of just 5.6 though, they're actually quite similar this year compared to previous years, around 250. And then the outflow concentration for 5.6 has been trending downwards. And so far this year, that inflow concentration is a little bit less than last year, so we do see a little bit lower percent reduction. Looking at the monthly trends, one thing that always sticks out is the frequent dry out conditions during the dry season. So you can see these gaps in the data lines here during those dry season months. And then following those dry out conditions, we do see elevated inflow concentrations and outflow concentrations for this STA. So in recent years, we've been working to hold that water in the STA as long as we can, as long as conditions allow, let biology do its job before that water gets discharged. And I want to add too that the severity and duration of these dry out conditions are expected to be decreased when the C-139 FEB comes online. For operational restrictions, pretty minimal in STA 5.6. There were a few periodic restrictions in flowways 2, 4, and 6. This was for some migratory bird nesting in flowways 2 and 6, and then a prescribed burn in flowway 4. I think Eric's going to talk a little bit more about in his presentation. All right, this is my final slide. It's a summary slide comparing the individual STAs. The charts at the top go from water year 22 on the left through water year 24 on the right. Then the table at the bottom shows information for all of the STAs combined with the flow volumes, loads, and concentrations. So as I mentioned throughout the presentation, so far in water year 24, we're seeing it's a pretty wet year, so higher inflow volumes, even with it being a partial year relative to more recent years. The inflow concentration for the STAs has been quite a bit lower for most STAs, and then the outflow concentration has been trending downwards. And finally, the percent reduction when comparing all the STAs combined has been comparable in recent years. And that's it for me. Leave my contact information there and pass it back to Robert.
Thanks, Jake. Great presentation. Our next presentation will be Eric Crawford. Eric is a lead scientist with the Land Resources Bureau, and he'll speak to us regarding the vegetation management process. Right?
All right, thanks, Robert. Vegetation management. Somebody once asked me why. You know, you don't get HR issues with veg, and there's nothing to do. I wish that was more true than it isn't. This is South Florida, so you could build an STA and get it wet, and plants will grow there. But they won't necessarily be the right plants or the plants that can do the job and take the phosphorus. So we hope that that happens, but then we got to get involved to tweak this system and make sure that we get the right plants growing. So the management objectives for the plants are to maintain the phosphorus uptake mechanisms. It sounds pretty simple, and for the first 70%, it probably is. After that, we've got to tweak it to optimize that performance. Lots of plants grow in the STAs. They don't all perform well. So our job is to enhance and maintain the appropriate plants in the appropriate communities, and we have to work to maintain the stability and keep functional redundancy throughout the flowways. Some of our communities that do our best phosphorus uptake, especially at the bottom end of the cell, they require protection. We have to plant other plants with those communities to protect them from wave, wind action, and from the fish and gators and other animals that are out there. So we do this through a lot of selective management. We try to increase the populations of desirable species, and we try to control the nuisance populations. We get a lot of floating aquatic vegetation coming into the SCAs, so we have to control that. We get populations of plants that aren't native here, that may flux phosphorus, that might die out, that may provide, habitat for bird roosting, which will give us a nutrient input. So we try to keep everything kind of in balance. The STs are big, so we can't work everywhere, but we try to identify the weakest areas and do what we can to help them. So a little review about the function of the vegetation. When an SDA is just starting up, either coming back from a dry season dry out or just starting up post-construction, the most important thing to do is to stabilize the soils. When the water first comes into an SDA, erosion is a big factor. So the first job of the plants is to stabilize the soils, to keep the bottom on the bottom, and to stop any preferential flow paths. We don't want short circuiting, we don't want erosion, you don't want gully formation right off the bat. After that, we're looking at maintaining sheet flow. The water comes in from a point source, spreads through the spreader canals, and then we want it to flow evenly across the cell. To do that, we have to have kind of equal resistance to flow across the cell. So you can't have weak spots and strong spots that will help channelize the water. So once you can decrease the velocity of the water and spread the flow out, we're starting to decrease the turbidity. You drop out a lot of the suspended solids. That plant growth creates litter. The litter serves as a source for the microbial action that can help pick up the phosphorus. And we also use the emergent plants to protect the SAV. So in areas of the cells where we're using submerged vegetation, we found that you cannot have a pure submerged aquatic vegetation cell. You need the emergence in there to protect the submerged plants from the water, from channelizing, from the wind and wave action. If you have a wide open SAV cell and tropical storm comes through, the SAV will be sitting on the levee. So we're trying to find the balance of emergence to mix in with the submerged to protect them while still maintaining enough surface area of the submerged to get the performance. Is a little different in every SDA. A lot of it depends on the flows, the soils, and just their orientation to the wind and how that cell catches water. So the submerged aquatic vegetation, while it's, I talk about how fragile it is and it needs protection, but it's where the water column nutrient uptake takes place. And it also serves as a really good substrate for the periphyton and the algae that grow on it that also assist with performance. So these two slides, this is a desirable mix of plants. So we have open water areas that are well colonized with submerged vegetation. They're divided by emergent strips and little islands of emergent plants that we use as nursery areas for when something like this happens, where you get an eroded channel. We can harvest plants from one of these islands, put them in the channel, and go back to a balanced flow. This slide it's almost all nuisance. I don't know if you can see the little patterns here. They almost look like waves in the vegetation, and those are created by pressure of the water moving. The soils in the cell had separated from the bottom and floated up. So all these plants are floating, and you almost get like tectonic plate formation where these mats of floating peat and muck and vegetation run into each other. So there's a process to get something from this to there. First you identify the healthy areas, and we mark them out, and then we have to sink the floating plants, remove the nuisance vegetation, and then do something to try and push back to a more desirable mix. Like this is a desirable SAV cell, and EAV cell would be a lot thicker. So the process of making those changes, it's it's kind of dependent on observation and monitoring. We've got people out every day trying to look at every cell, but we've got a lot of acres to cover, so. And we need to proactively manage the vegetation. You don't want to wait until something dies out before you go in and fix it. If we see an area that looks weak, we need to get in and stabilize it before it fails. Because if it fails, then you have a drop in performance, and we have to fix it and then get performance back. So we try to not have the failures first. And a lot of that is just maintaining the cover and maintaining the health of our desired species. And for a lot of that means removing the undesirable species. We use cattail in the emergent cells for the majority of the vegetation. It's almost always a desirable plant, except when it's not. Cattail doesn't root very deeply. It can tend to separate from the soils and bring the soils up to it when we have flood control operations and rapid changes in stage. Cattail can peel off the bottom. Once it's peeled off the bottom, it's catching wind and it can get mobile. Those mobile islands of cattail can destroy other plants and mow other healthy areas down. So we get in there and we put in deeply rooted species. We mix them with the cattail. We support the cattail. And then we just restore the damage, or repair the damage and restore the functionality. Short circuits form, we have flood control, we have wildlife, there's all kinds of impacts on the soils and the plants, and we try to catch those things as early as possible. It's really hard to get plants growing in an established short circuit where the soils are scoured down to mineral soil and there's heavy flows and it's fast. So we try to catch it early, plant it while it's still plantable without a drawdown. So this picture's it's an old one, but it's one of the best pictures we have showing how the plantings are done. We float in rafts or bundles of plants, and they're put in one plant at a time by hand. So obviously we can't plant the whole SDAs. We plant a tiny proportion and try to put the efforts where they're needed. Normally, we're not planting a big area like this, but this was a mucky zone where the soils were continuously delaminating. So we planted this with bulrush, which roots really deeply and helps to stabilize the soils. So once we can get these populations thick and get this physical structure at the front end of the cells, then we can go back towards something more like sheet flow with balanced water movement across the cell and the flowway, and that can't but help the performance. So SAV management's a little different. We don't plant individual plants. We inoculate with plant fragments. Sometimes we plant individual plants, but for the most part, it's inoculating by fragments or bringing in propagules. We learned the hard way early on that the SAV cells need to be compartmentalized to protect the SAV from wind and wave action during storms. We found that smaller and more diverse and more compartmentalized SAV beds are more resistant to flow, more resistant to perturbation, and they're more resistant to seasonal die-offs. A lot of our SAV species will senesce during the winter. Not every winter, not every species. We're not in Pennsylvania. They don't, not every fall they don't die out and come back. So by trying to mix the species, we prepare for senescence and we prepare for recolonization. In different areas and different soils, it takes a different mix of plants to get the colonization back. We try to have it ready so we don't have to get involved when it happens, but we have to keep it under observation in case it needs help. So FAV, it's something I complain about a lot. The floating aquatic vegetation, It gets bad. It comes in, it can come in during a storm event. The pressure behind it can be enough that it'll bowl over the cattail, and it'll lay the desirable vegetation down. It can shade out the SAV. It's bringing in a lot of nutrients that aren't counted. You know, they're big particles, not something that makes it into a water bottle. So these rafts of FAV come in, and we need to stop them and protect the desirable vegetation. Some of that's done through herbicide application, but for the most part, we try to hold it back. We try to plant the emergent plants in a pattern that'll hold them in the front end of the cell and keep them from ever making it to the submerged aquatic vegetation cells. So we know we're getting FAV in our A cells. And we've instituted a lot of upstream controls trying to reduce that amount coming in, and it's helping a lot, but it still comes. There's no stopping it. When a storm comes, the water moves. You can see in this picture, these brighter green areas are all submerged aquatic plants. That's all water lettuce, and this is hyacinth. So 2 different species of invasive FAV that have come in and that are threatening the cell. The pressure from the FAV can actually hit rooted cattail, and if it's on a thin, mucky soil, it can uproot the cattail and cause more floating tussock problems, which are difficult to remedy. So SDA 5.6, I think Jake mentioned we did a controlled burn out there. We have regions of 5.6 because of the past operations and the seasonal drawouts that are heavily colonized by woody species and undesirable and nuisance plants. We know that a burn can help reduce those, especially the invasives. And we also wanted to reduce the standing and horizontal dead cattail biomass. And some of these areas and the way that the flows and the stages fluctuate throughout the year, the cattail became really thick where it was healthy, but it gets too thick, and with the dry outs, the dead biomass isn't decomposing. And so it builds up, and it gets to the point where, you know, I'm not really light, but I can crawl across the dead cattail leaves without touching the water. And when the cattail's dead biomass is strong enough to hold me up, it's also horizontal, so it's almost like a beaver dam across the cell. So it causes a great resistance to flow when the water comes in. You resist the flow, the pressure increases, so it either goes to a weaker spot and scours it out, or it bowls it over and it takes the live cattail with it. So we tried to reduce that dead biomass with the fire, and it also helps to increase light penetration. If you do it the right time of the year, the light can get down to the water, so you can even get SAV in there, or if it's an EAV cell or a place that's gonna dry out, when you can expose those soils and give them light, you can get better seedling success and performance. So you can see the fire line going right there. It was a nice controlled burn. And this picture was 3 days after the burn. You can see this burned out area here, really brown. This area burned really well. There were a lot of willows and primrose willow in here. So a lot of this burned out right down to the water level. 5 weeks later, it's almost all cattail. So the recovery post-burn was really nice. We're still working on the numbers. And you see there's kind of a weird pattern in the burning because some of it was prepared before the burn and some was not. We used a contact herbicide to kill off the standing cattail leaves so that they would burn more quickly, which helps the fire build energy and move across the cell quickly. Some things could have been done better. We didn't get the burn as quickly enough after the treatment as we would have liked, but, you know, burns are dependent on a lot of factors. We had to have the water available to put the fire out before you could start it, and we're restricted on wind direction and strength. So you have to wait and have the crews ready until conditions are appropriate. But so far, we're really happy with how it went. It hasn't been long, and we haven't seen a wet season post-burn yet, but I'm pretty happy with the way it looks. So erosion, I talked about at the beginning. These SCAs are out there for water quality improvements, but we can't always trickle flow or flow at a nice low rate for where you need, you know, where you would maximize your performance. And you get a weak spot in a cell like 1E cell 2, which has a sandy bottom. Storm operations cause a little bit of erosion. It just takes one weak spot to start a short circuit. The short circuit starts, the water picks up velocity, storm comes in, operational changes happen, and the water moves more quickly, and we end up with water flowing not necessarily through the marsh, but around it and right to the outflow region. So this is what that area looked like when you take the water away. The outflow canal was filled with sediment. Soils that should have been inside the cell were now in the outflow canal. The picture on the left, you know, this should be about 6 feet lower than it is. The birds really like it when we do a drawdown, and they show up. The fish all concentrate in the canals, so the gators and the fish show up and feast during the drawdown. This just shows the extent and the depth of that short circuiting. It can start from a tiny little channel or something that's a 1-inch difference, and then a weak spot in the plants, and now we have problems to fix. So the construction group is in there. They're digging out the canal. They're putting in a generator so we can fine-tune storm operations, so hopefully don't have that happen again. And you can see veg management was in there. We had crews in there planting at the same time as the construction guys are working. They have their little buffer zone, we stay out of it. And we're trying to work in there simultaneously with the erosion repairs. So as soon as they get the earth moved, we can be planting and getting things ready so that when we can bring water back into the cell, we can get back in operation quickly. This is a really well-performing cell. It's an SAV cell, so we don't want to keep it dry for too long. Hopefully, we'll have water back in there by May, I hope. And we'll be able to get some good growth going this wet season. And, you know, you never know what you're going to find in the STAs. We've had manatees. We have a bull shark here. They're beautiful places. I know it's a water quality improvement facility, but there's a lot of wildlife out there. The birds are amazing. The plants are great. Obviously, I like the plants, but you know, come out, go for a walk, look around, and that'll do it. Thanks.
Thanks, Eric. That was very good, especially the bull shark and the STAs. The next presentation will come from Dr. Tom James. He's going to discuss the status of our RS science plan. So with that, I'll yield it to Dr. James, Principal Scientist of Applied Science.
Thank you, Robert. As he mentioned, I am Tom James. I'm the coordinator for the Restoration Strategies Science Plan. I did want to mention that this science plan will be concluded this year. So we've got a lot of work left, but when I begin this presentation, I'm going to give you a background of the restoration strategy, science plan, the RSSP, provide a list of studies and what we've done in the past, and focus in on some of the detailed, completed and ongoing studies, and finally end up with some of our outreach to the public and scientific community where we've presented our results. And let's see if I can do this right. Good. So the science plan itself, developed in 2013 and updated in 2018, is specified by the Restoration Strategies Regional Water Quality Plan. Required by 2 different permits, the National Pollutant Discharge Elimination Systems and the Everglades Forever Act. The concept is to provide some understanding of the processes that affect pee retention in the STAs so that we can use that information to optimize the system and result in improved management of the system to reach the Q-belt, which is the water quality-based effluent limit. Now, the whole system was based, or the plan was based on areas of investigation, and these were geared around 6 different topics that include the design and operation of flow equalization basins as well as STAs, and looking at vegetation management and maintaining sustainability of vegetation as we just saw a few moments ago. Then looking at internal loading of phosphorus and the biogeochemical and physical mechanisms that are creating the changes in phosphorus throughout the STA. And finally, the role of fauna. Now, it's taken a team to do this of scientists, engineers, field crews, technicians from not only the district but also from our private consultants as well as universities. And they did hundreds of experiments and provided hundreds of reports, thousands of samples, and hundreds of thousands of data points. So I just wanted to make sure that when I use the term we, I actually mean they, because I'm just the messenger today. So as of 2023, there have been 15 completed studies. All of the studies dealing with hydrology are in light blue. And let's see, light blue. Then those that look at soil-water interactions are in brown. The dark blue are the biogeochemistry studies. And then the green are plants and plant-water interactions. And finally, we look at these orange ones are mostly monitoring and analysis. We still have a few studies left, 6 studies to be exact, and 5 of them, plus the 15 completed studies, will all be basically pulled into the data integration study, which is the final piece, and I will discuss that in a moment as well. I wanted to begin with the L8 flow equalization basin study, which was recently completed. This one came about because this L8 FEB at one point, this is water year 2018, the outflow concentrations were greater than the inflow concentrations. And what we really wanted to know was what's happening, where is that extra phosphorus coming from? So the objectives were simply to determine where the sources were and to develop some sort of guidance for reducing TP in the discharge. So one of the things that we did find, we looked at various possible sources, groundwater and runoff from embankments. We found those were very minor. But we looked more to the major large inflows. And here's the slough that runs into the L8 FEB. And you can see that this large amount of water going into a low-level L8 produces a fair amount of resuspension from the sediments. So this actually caused a lot of phosphorus to go into the water column. One of the things that we do see that after such an event occurs, the phosphorus does decline after a few weeks. So one of the other things we learned through this particular study was that TP is strongly related to turbidity and dissolved inorganic phosphorus, and we've actually been able to use that for some guidance here. In addition to this whole set, we said, are there any other things we can do to reduce phosphorus in the FEB? And we looked at an alum feasibility study. Now, alum is a chemical that's used in sewage treatment and water treatment to essentially create flocks of all sorts of material to get rid of it, including phosphorus. It's also used in lakes that have been eutrophied, that have been have too much nutrients, and it actually does remove the phosphorus pretty well. It's been very successful throughout the nation and throughout the world. So at low concentrations, a fairly low concentration, the alum does remove organic P. And what we did with this feasibility study was to look at potential for developing actually a whole construction site of pull together the site to deliver alum to this slough to see how much it would cost over time. So we did some jar tests. This is basically putting some water and putting some alum at different concentrations to see how much phosphorus was removed. And then we ran some model results. And basically the model results showed that there's a small amount of P is removed by the alum, and it's expensive. It's estimated right now as $4,000 per pound. That may actually be reduced as we look at this a little bit further. So this brings some potential options, essentially from what I mentioned before. If we retain the water after an event for a few weeks, the phosphorus levels should go down. The other thing that we've been looking at is using near-time monitoring of DIP and turbidity using some sondes. Here's a sonde that actually measures DIP in the water column, and put this close to the outflows. And using that and turbidity, we can estimate phosphorus concentrations at this region, and that provides some estimates of discharge from the STA to the STAs. And this can be used by our managers to basically determine what the phosphorus levels are and when they can discharge, or best times to do that. Another study is the Marl study, and Marl itself is essentially produced it's produced based on photosynthesis of the SAB, which causes a chemical reaction to create calcium carbonate, which is a precipitate, and this material gloms off phosphorus and sinks to the bottom. And as you can see, here's some of this marl that's created, actually the calcium carbonate, and then it collects in the bottom of the soils, and it actually collects fairly large amounts. And then finally, it tends to be fairly easily resuspended. Here's a fish that's moving through it, and it looks like it is, it's really, really easily resuspended. So what we're concerned here is to look at the marl soil stability, trying to figure out ways of increasing the stability of the material and reducing the phosphorus coming back into the water column. So we will look at two potential methods to reduce resuspension. One is drying, just taking the water out of the soils and making it more solid. The other is to add organic materials, which might be kind of a glue to keep the soils together. So we did a couple of studies using some laboratory tests of cores, and we found that marl soils is actually less susceptible to resuspension than organic muck soils, which was interesting. So it may not be as much of a problem as we thought. We did find that the organic materials themselves, when we added them to these cores, they didn't help. They weren't very effective. And drying followed by rehydration, actually for a day or two, was very effective in keeping the flux of phosphorus into the water column low. But after about a week, it increased to more than our controls. And then finally, after a few weeks, it came back to normal. So that's not a good thing. But we also note that, and you saw something that Eric showed you before about the SAV and dryouts. These conditions do occur in the STAs. And if a cell does dry out, we do have a condition where the SAV will germinate relatively quickly, within a few weeks. So that if the cell does dry out, once it's reflooded, we want to retain the water for a few weeks to allow SAV germination. And this was from the SAV study that was previously completed. So one of the things that we do know about the phosphorus that comes into these STAs is that it really, most of it is retained, and some of it is recirculated and recycled through the system. So the question is, where is it going to and where is it coming from? And this is where the biomarker study came in. We wanted to analyze organic material as markers of the phosphorus origins and to determine the sources of potential turnover within the STAs. And the first thing that we looked at was the inflow waters. And as is fairly obvious, the inflow waters themselves are different based on watersheds. Their dissolved organic matter is different. One of the sort of interesting pieces that they found in their investigations was that exposure to sunlight, also known as photodegradation, will break down this dissolved organic matter as well as the dissolved organic phosphorus that's within it. And that material is easily uptaken by the microbial community. So it actually may be a benefit to have areas where there are exposed water column to allow the sunlight to break down some of this material. And this is something that, of course, occurs because we do have lots of SAV out there. And you can see here a lot of area that is open and lots of sunlight can get in. So we also wanted to look at fresh litter, and we came up with these litter bags, very small kind of tea bags almost, and put litter in them and put them out at into the STAs for different amounts of time, 0, 20, and 40 days. And then we put them into teacups, essentially, and let them set to see how much material came out in dissolved organic matter. And in fresh litter, if it's real fresh, 0, just cut, as expected, the DOM released very quickly. For SAV litter, it's much faster than EAV litter, and with the SAV litter, there's a lot more dissolved inorganic phosphorus that was released from the SAV. In terms of DOM in the STAs, we do see that it's primarily from this litter decomposition. So this is where the source is maintained. It goes through the plants die, and the microbial community and all sorts of other activities create the DOM, and that gets into the water column. And the microbial processes toward the outflow area really process it and affect it more than sunlight at that point. So one of the potential options here is to maintain those open water areas in regions of the STA to allow photodegradation of the DOP. The fauna study, which is ongoing, one of the interesting things that we found early on is an observation of these fish nests, which are down in the STA near the outflows. And you can see that they've created quite a lot of nests, a lot of the soils have been disturbed, and they've actually replaced a lot of the SAV either through basically burying the SAV or maybe some of them herbivory throughout this. So what we wanted to do was quantify the fish and the effects on the phosphorus cycle. And one of the things that we did was look at the large fish densities and small fish densities. And we found that the large fish densities were related to water levels. As, again, Eric mentioned before, that At low water levels, the fish tend to be in the canals, and that's where all the birds and the alligators can feed, which might be a good thing if we look at it. But we also said that our methodology, which is this method using electrofishing, you can see this is our setup here. It tends to underestimate the amount of fish, and we actually went out and did some experiments using some big enclosure nets and ran this electrofishing apparatus through it to collect the fish, and then we went through and picked up the nets and found out what was left, and we found that the capture rate was between 1% and 50%. So when we used that to essentially as a fudge factor to improve our estimates of fish, we did see that these fish do create, they're fairly large amounts of fish out there, and that they do substantially recycle the phosphorus. Now, large fish mostly through this bioturbation. Here's our setup for the bioturbation experiments, where we did put fish in a couple of these enclosures, and some you can see where they're really, really turbid. That's where the fish are, and the ones that are clear, there's no fish. So they do create a lot of bioturbation, but it depends upon the type of fish. Sailfin catfish itself is very high bioturbator, versus largemouth bass, which really doesn't go into the soils, is not. Despite that bioturbation, the small fish, which are in a large amount of biomass within these STAs that we determined through throw traps, actually their excretion was about 3 times greater than the bioturbation from the large fish. So all of this excretion and recycling of phosphorus impedes the phosphorus retention. That doesn't mean that it's an impediment, but it doesn't mean that we're reducing, that we're removing the phosphorus, or we're still removing a lot of phosphorus. Somewhere between 80%, I think, is the last year's estimate. Another estimate thing that we looked at was herbivory, and this one is just being completed, this experiment. And you can see here's a control. This actually is has openings here, versus this one is closed out and no fish in it. And you can see after 3 or 4 weeks, this SAB has regrown from bare soil. So the herbivory report is still in review, and essentially to regrow SAB in bare areas, we could lower the water levels if possible. Again, this does occur upon occasion, to reduce our fish herbivory of growing plants. So one of the other, excuse me, one of the other species of plants that we look at is the periphyton. These are the microbial components that exist in the STAs. Here we go. And they're hard to measure in terms of their processes. So we looked at a couple of different things to quantify their role using metagenomics, which is a DNA, go into the CSI types of things, and microscopy to see essentially what's there, and also to get an estimate of sorts of the processes that might be existing, including those that are operating, the different phosphorus metabolism enzymes that are active based on their RNA, based on the RNA itself. So we did also some lab nutrient addition incubations to do the same sorts of things, look at the metagenomics and the microscopy, and a field flume study that looked at the processes of different flows on essentially creating turbidity within all of this plant life. So right now it's just, we've looked at the metagenomics, and there are seasonal changes. So there's changes in the population of the microbes in the community, and there are changes into the different genes that are turned on and off. So the status now is that the sampling's complete. We're still waiting on all of the different studies to complete and be analyzed. So early on in the restoration strategies, we had a study called the P-flux study, which looked at well-performing phosphorus, well-performing STAs in terms of their phosphorus removal. And we wanted to look at the same type of study looking at underperforming flowways. And one of the things that they found was that underperformance, just based on our historical analyses, were essentially based on a couple of different big items. One is high external phosphorus loads, dry out reflood, damage or loss of vegetation, and construction. If you didn't have any of these in an STA for a period of time, you get really good performance. Now, if we looked at the soils and you wanted to see if this was same as it was in the P-flux study for the P-dynamic study, it was. There's consistency here in that the EAV, they have mostly organic soils, and the SAV is mostly inorganic, this calcium moral. Now, one of the things that we've noticed throughout of our studies is that under no-flow conditions in SAV regions, the water column is dominated by particulate phosphorus. And also, when there is flow, and if the flow is low, the TP concentrations tend to be higher. Again, this is very consistent throughout most of our studies. So right now, all sampling is complete for this particular study. We're waiting for the analysis. So this goes to some of Eric's concerns, is what plants and what plant communities essentially provide the best P retention, especially in the outflow regions of the STAs. And we wanted to estimate the phosphorus retention by the common STA communities or ecotopes, which is the community plus the current conditions, the chemical and environmental conditions that they're exposed to. One of the first things that we had to do was to find communities that were maintained throughout time. And these are very dynamic systems, as we have seen by Eric's presentation. But you do see here's two of the same area, different seasons, and you can see there's lots of differences here. But to look at these particular ecotopes, we basically went in and sampled them on a monthly basis and provided that information to rank the different ecotopes by their TP concentrations. So, of course, the one thing that's come out, and this is again very consistent with all of the other data that we have, is that Cara is essentially the community that has the lowest phosphorus concentration versus the Taifa and the bare soil at the top. Now the concern, or the interesting thing with this, is that these differences between the communities is smaller than the differences by season. But consistent with a lot of the other studies is that DOP is the largest fraction. The other thing is, of course, with higher flow, as I mentioned before, we have lower TP concentrations. We're also looking at the SAV genetic diversity, and this is an interesting concern because we were thinking that there might not be a lot of diversity in the SAV, and that could lead to some problems in the future of the community being stable and resilient. But we found thus far that there's been high variation in the genetic diversity within and between sites where we've actually sampled. And there's little clonal reproduction. So this is a positive thing, and we think those communities should be more resilient because they are very diverse in terms of their DNA genetic diversity. So the sampling is complete with this one. We're still waiting on final analyses. So one of the studies that we've done in the past is the landscape study. This is part of another study. And we wanted to quantify flow and hydraulic mixing given different plant densities. And they actually tried to do this at the STA cell level in the past. And they did have some information that was provided, but we wanted to see if we could do this in more controlled conditions. So we created these two flumes, a straight plume and a V-shaped plume. And one of the nice things about a V-shaped plume is that when you put water at the top, the flows tend to reduce as the distance between the walls increase. And this allows you to look at multiple flow rates at the same time in terms of mixing of the water column. So what we're doing is evaluating multiple flow velocities at once again, and looking at the baseline. We've actually done this. The baseline was looked at steady state flows. We looked at salt and phosphorus that we added to see where the mixing was occurring. And finally, looking at some standing waves to show us some of the resistance that might occur. Now, the cattails were planted back in October, and you can see here's what they look like in the past month. And the first round of experiments are actually out there this today to complete this. Oh, sorry. So we get to the final piece, which is the data integration study. We're going to compile all of the information from the Restoration Strategy Science Plan, develop potential options to optimize the P removal, and determine gaps in the research. And this will come out as a final report. We do know already that low phosphorus outflow is associated with phosphorus loading rates lower than 1.3 grams per meter square per year. There's no disturbances. There's dense SAB, active periphyton community. The biogeochemical and food web model we're actually producing to give us a little bit better information or some ideas of what's going on within the STAs. And that's being developed, and we're waiting the final calibration of that. To synthesize the results from previous prescribed burn study is also being done to help support veg management. So finally, we've got our biogeochemical and food web models, which are being validated, and we're creating this final synthesis document. Now, we do have an outreach. We've been doing some outreach in the past year. We have the South Florida Environmental Report. We've presented last year all of these different studies, and we also presented 3 posters, which were included in the poster session. There's going to be another one this year on April 6th. We want to invite everybody to that, and we will be participating with updates of these particular posters. We also were present at the Greater Everglades Ecosystem Restoration Conference back in April of last year, where we had 29 presentations and 7 sessions. We were very well received there. And finally, there was an ecological engineering special issue that we are putting together. 18 of these 20 presentations or 20 manuscripts have been published. There are 2 that's still under review. But once that's under review, then these will be in open issue, may open access, and will be available to the public. And with that, I am ending. So thank you very much.
Thank you, Dr. James. Very good presentation. Lets you know we're doing a lot of activities, a lot of research that's going on. Our next presentation, our next presentation is actually looking at our engineering and construction update for the restoration strategies. And it's going to be done by Mr. Alex San Miguel, who's the project manager and the section administrator in Engineering and Construction Bureau. So I yield the floor to you. Yeah.
All right. Good morning. My name is Alexis San Miguel. I'm a project management section administrator for Engineering and Construction Bureau, and my presentation is not going to be as interesting as the science that you have seen. It's a little different, but it's good to hear all those. Since I'm in engineering and construction, just hearing about all the background on the science, I get a refresher on that. So the projects that were listed on the restoration strategies were divided into 3 different flowways. And it was to help on the improvement on the system to meet the QBL. The 2 projects that are left to be completed construction are this little triangle here is the STA 1 West expansion number 2. Is on the eastern flowway, and then on the western flowway we have the C-139 FEB. This slide shows all the progress that has been made, which basically all the construction of all these components, STA2 expansion of compartment B, the compartment C, S5S modifications, the L8 FEB, A1 FEB, which you have been hearing about these projects already in the previous presentations, and other smaller components. And this one is on the optimization phase, STA 5-6 earthwork. You're going to hear a little bit of expansion number 1 of STA 1 West on this presentation. Also, G341 segments 1 through 5 was recently completed. Some people know it as a Bowles Canal. And then STA 1 East repairs. So the 2 projects that are currently under construction in the later phases of construction, we expect them to be completed this year, are the 1 West Expansion 2 and C-139 FEB. So STA 1 West Expansion number 2 is part of the STA 1 West Expansion project, which consisted of expansion number 1, which is here in blue, and expansion number 2, which is this little triangle. Expansion number 1 was completed 2018-2019 timeframe, and the expansion number 2 project, it's around $240 million and around 1,600 acres of additional treatment area. Between the 2 expansions, it added around 6,500 acres of additional expansion. This project helps with the inflows of the S5A and C51 West basins coming through the STA 1 West and then into either of the 2 expansion areas. This expansion number 2 project is connected by a concrete line channel. It's a 6-mile channel here. Expansion number 2 consists of 3 cells, cells 9, 10, and 11. And like I said before, it's around 1,600 acres of effective treatment area. This project was divided into 4 different contracts. One of them was an underground piping project that was completed back in 2020. It was to connect the inflow pump station into the collection canal here to go under an existing canal. Then the STA and the collector canals, like I said, is at the later stage of completion. The project is actually the STA component and connector canal are substantially complete at this moment. But we still have to complete final completion of the project. The inflow and outflow components of the project at the pump stations, I'm gonna go back to the previous slide. That inflow pump station, one of them is up here to send water that have been treated already from the yellow triangle, send it south through the canal and get it to the expansion number 2 area. That second inflow pump station is to pump from the connector canal into the SDA. And then the outflow pump station, it's to send water into the water conservation area. Here you can see a couple of pictures for the progress of the project. The concrete conveyance canal, it's this over here is a 6-mile canal lining concrete adjacent to the water conservation area. And the water flows here until it gets to the 781 pump station, which sends water into the inflow canal of the SDA. Here's a picture of the inflow canal on the north end of the SDA and the seepage canal on the north to protect the lands on the north. And here you can see already some water inside the cells. Here you can see the G780 pump station. This here is cell 8 of the expansion number 1 area. But you can see already there was a plug here. It was already opened. And water should flow under this canal into the connection canal here. Then the 781 pump station is the one we saw in the earlier, but this is more of a close-up, so you can see the progress of the construction. This is the outflow pump station, which pumps into the water conservation area, is right here. There's still this plug. Actually, this plug is planned on being removed in the next month. There's a plan on how to get that done. But this, you can see the pump station is well advanced, and progress is being made, and we expect it to be completed this year. Next, we have the C-139 flow equalization basin. This is located west of the SDA 5-6 and north of the C-139 annex. And it will take the inflows from the basin and the discharges to the SDA 5-6 to provide the attenuation and reduce the dryouts that others spoke about earlier. Okay, so this project also is on the, it is still not substantially complete, but it's getting there in the next couple of months. For the overall project, it has a inflow pump station on the northeast end. It pumps into the FEB, and there's an internal levee that goes, but it's a partial. It doesn't get all the way to the end. It goes here, making the water travel a long path, go back, and then the outflow pump station, it's in around this area here, and then it pumps out here. There's also a seepage pump station in this area here, And like you can see here, they're plus 90% completion on all these features. So this project will be getting into the optimization phase soon. Just to give you some more information for people that are not aware of this project, it provides around 10,500 acre-feet of storage and a maximum storage depth of 4 feet. The discharges go into STA 56, and the inflow pump station has a 690 CFS capacity. And let's see a couple of pictures of the progress. This is the inflow pump station, and this is the 551 outflow structure. You can see that it's pretty much getting to the end of construction. This is the seepage control structure and the 711 east control structure. These pictures are probably 3 to 4 weeks out, so it's probably in better shape that the picture shows right now. This slide shows the components of from restoration strategies. You can see expansion. What's left to be checked are the completion of construction of the expansion number 2 project and moving into the optimization phase. And on the western flow path, the flow equalization basin that we just went through. And that's what I have here. Here's my contact information, and thank you.
Thank you very much, Alexis. One of the things that he was talking about specifically, that list, that final list that list is literally 13 years of work that included 13 projects of the restoration strategies. The fact that we're down to 2 is obvious that it's all coming to close, and that's something to really celebrate. Our next presenter is Dr. Yochao Wang. He's going to talk to us. He's going to talk to us about the Southern Everglades nutrient source. There we go. Nutrient source control program. Dr. Wang is a lead engineer with the Everglades and Estuaries Bureau, Protection Bureau, and I'll yield the floor to Dr. Wang.
Thank you, Robert. Good morning. My name is Yochao Wang, and I will provide you with the update on the source controls in the basins discharging to the Everglades. Source controls for this presentation are the activities or projects to reduce phosphorus before it reaches to the stormwater treatment areas or receiving water bodies. A typical example of the source control is best management practices, or BMPs. These basins this map shows the basins tributary to the Everglades. So on the map on the screen, the green area called the Everglades Construction Project basins. These basins discharge the stormwater runoff to the stormwater treatment areas, or STS. And those in brown, the basins also called non-Everglades construction or non-ECP basins. So these basins can discharge stormwater runoff directly to the Everglades. This slide depicts how the long-term plan and the restoration strategy supplement the mandatory regulatory program of 1994 Everglades Forever by providing funds for additional projects and describing comprehensive strategies. So the goal of the 2003 long-term plan is to, by identifying cost-effective opportunities and working in partnership with the landowners and municipalities. So today's update will describe the source control program in the Everglades agriculture area, the called EA Basin, show the orange in the map, and also C139 Basin. So these basins, the source control program are implemented under the mandatory program chapter for the E63, and also for the other tributary basins shown in the brown in the map. So this basin can discharge directly to the Everglades. So the source control program for these other tributary basins are implemented with the combination of regulatory and cooperative efforts, and ultimately rely on the comprehensive Everglades restoration plan project or sub-projects. This table provides the overview of the water year 2023, the receiving water body, the water year 2023 TP load value and TP flow with mean concentration for each basin. The basins are sorted based on the TP load values here, from the largest to the smallest. We will provide a quick update and summary for each basin except for C111 basin, which is not included in the long-term plan. And more detail is provided in the 2024 South Florida Environmental Report. Let's start with the largest tributary basins, the Everglades agriculture area, the EA Basin. So for the EA Basin, the source control program includes the EA Basin level water quality compliance, the permit level compliance, research and demonstration project, and the sub-regional source control project in the eastern flow path located in the S5A sub-basin. So this slide shows the EA Basin level compliance with the 25% TP load reduction requirement. So the bar here shows the annual TP load reduction for each year from water year 1996 to 2023. And the dashed black line here is the 25% TP load requirement by law. And the green line represents the long-term TP load reduction. So the EA Basin level compliance is, the EA Basin is determined to be out of compliance if the 25% TP load reduction is not met for 3 consecutive years. This is for 3 consecutive years. Here we can see like in 2007, it's below the 25% TP load reduction, but this is just 1 year. So if they have 3 in a row, the basin will be in compliance, will be out of compliance. So for the EA Basin, the EA Basin is in compliance since the program started in 1996. And for water year 2023, the TP load reduction is 63% with a long-term reduction of 57%. This slide shows the EA permit level compliance. So for the works of the district permit, that requirement include implementing of the comprehensive best management practices or BMP plan, and also the permitted water quality monitoring plan and post-permit compliance activities. So the BMP plan, which means implementing the nutrient management the water management, and particulate matter and sediment controls. So these pictures just show some example of the BMP implementation. Stepping into the research and demonstration project. So for the EA basin, the Everglades Forever Act require implementing a comprehensive program of the research, field testing, and the implementation of BMPs. So a 5-year EAA EPV master research permit was issued in September 2020. So this research is to evaluate the performance difference between the EA farmers with similar BMPs. So as shown in the map here, 3 pairs of total 6 farms were selected for this study. And the third interim annual report was completed in July 2023. The sub-regional source control project is to supplement the existing regulatory best management program. So the intent of the sub-regional source control project is to reduce phosphorus in the eastern flow path. So eastern flow path, basically primarily in the S5A subbasin. So currently there are 2 sub-regional source control projects under consideration. So the first one is located in the upstream of the S5A pump station, West Palm Beach Canal dredging project to avoid sediment resuspension. So the second one is East Beach Water Control District load TB load reduction project. Moving to the C139 basin. So the C139 basin, similar to the EA, also implement the regulatory source control program required by the Chapter 46-3, and with maintaining the basin level below or equal the historical TB load level. So unlike the EA basin, the C139 basin did not apply for the permittee level water quality monitoring, but the district has established the subbasin monitoring and upstream monitoring. So the subbasin and upstream monitoring station are shown in the map here. This figure depicts the C139 basin level compliance. So for this figure, we see the bars, all these bars represent the measured TP load for the C139 basin for each year. And the yellow diamond represent the TP load target, and the red diamond represent the TP load limit for each year. So the compliance assessment for C139 basin is compare your measured TB load in the bar with your target TB load, the yellow diamond, and the limit TB load. So if the bar below the target that year, so the basin is in compliance. If the bar above the target for 3 consecutive years, or above the limit for any single year, so the basin is out of compliance. So please note for the water year 2018, the target and limit is high, is out of scale. So for C139 basin, so the orange bar that represents the basin is out of compliance, and the green bar represents the basin in compliance. So from here, we can see the C139 basin is in compliance since water year 2010, the last 15 years. Moving on to the other tributary basins. So our first basin we'll talk about is the C51 West and Acme Basin. So for C51 West and Acme Basin, so the C51 canal is bisected for the C51 West and Acme Basin. So south of the C51 canal is called Village of Wellington. that include Acme Basin A, Acme Basin B, and Pantry Water Control District. So historically, the discharge from the Acme Basin A and Acme Basin B can discharge to the refuge directly through these 2 pump stations. After 2007, the SERP, the Acme Basin B SERP project completed. So the Acme Basin B, the stormwater runoff diverted to Acme Basin A and eventually go to the C51 canal, and so can flow to the west to the STI1 east for the treatment. So for source controls, the BMP implementation are required through the environmental resource permits. So that the permits issued to the village of Wellington water control district. So the BMP, the water quality monitoring also required for this basin. So here the blue U mark represents the water quality monitoring in the village of Wellington location. And the green U mark on the north represents the north of the upstream monitoring site conducted by the district. So regarding the water quality data, so the chart here, the figure represents the Acme Basin A and B discharge to the refuge prior to the SERP 2007 project completed. So as we can see here, the bar represents the annual TP load. The dark blue diamond represents the annual TP fluid mean concentration. So there are no discharges after 2007 because of the SERP project, so all the water diverted to the C51 canal, except the 2013. So a small discharge to the refuge directly because of the village of Wellington is under emergency water control, flood control condition due to the tropical storm Isaac. So in that means under emergency condition, the village of Wellington can still discharge to the refuge. So after 2007, the stormwater runoff from the Acme Basin divert to C51 West Canal and to the STI1 East for the treatment. So the chart on the right represents the discharge to the STI1 East from the C51 West and Acme Basin. So the pink bar represents the annual TP load, and the green diamond represents the TP concentration. So for water year 2023, about 13 metric tons to the STI1 East. So our next basin is the Feeder Canal Basin. The Feeder Canal Basin is an agriculture basin located in the Hendry County. So the Feeder Canal Basin include the North Feeder Canal Subbasin and then West Feeder Subbasin, and also some land of Seminole tribe. So the whole basin, the discharge is through the spillway S190 here. So all the basin discharge. For the source controls, so the district continue oversight the BMP implementation through the ERPs issued to the landowners in the North Feeder Subbasin with the goal to manage the runoff discharge fluid mean TB concentration no greater than 50 ppb. That's the goal agreement. There are also 2 voluntary FIV tilling projects in the North Feeder Subbasin. For the West Feeder Subbasin, the landowners can enroll in the FDEX BMP program. So for the Feeder Canal Basin, the SERP project, the SERP Big Cypress L28 interceptor modification project rely on the completion of the Western Everglades restoration plan or WARP project. So the goal of the WARP project is to improve the water needed to for the quantity quality timing distribution of the water needed to reconnect and restore the Western Everglades restoration project. So the tentatively selected plan would achieve this benefit. So regarding the water quality data, so from here now on, I just say for all the charts are the same. So for all the basins, from forward, all the bar represent the TP load, and the dark blue diamond represent the TP concentration. So for Feeder Canal Basin, the annual stormwater runoff discharged through the S190 structure is shown here. So for water year 2023, about 10 metric tons TP load discharged to the Everglades with the fluid mean concentration of 121. So the L28 basin is also an agricultural basin located in the portion of the Broward County, Hendron County, and Collier County. So it includes 4 landowners. So the district owns the C139 annex. So this portion, the district owns this land. For the source control program, so the BMP implementation through the ERPs issued to the landowners, to the Southern Garden Grove, that the landowner. For the L28 basin, the third project, as we said before, in the Feeder Canal Basin is rely on the completion of the work project. The other projects include C139 annex flow equalization basin and San Juan Abiake Prairie restoration project. And the same, for the water quality for L28 basin, the same, the bar represents the annual TP load. The dark green diamond represents TP fluid mean concentration. So we can see the each year varies depending on the rainfall, stormwater runoff, and the program. So for water year 2023, about 10 metric tons discharged from the L28 basin to the Everglades. C11 West Basin is located in the Broward County. 70% of this C11 West Basin is urban area, with the rest of the agriculture. So this basin discharge the storm runoff discharge through the pump station S9 and S9A. For the source controls, the BMP implementation through the ERPs issued to the C51 West 3 water control district. They have the ERP conditions include the BMP implementation for optimized detention of stormwater runoff and water quality monitoring. For the third project in the C11 West Basin, the third Broward County Preserve Area project include 2 above ground impoundment C11 and C9, and also a wetland buffer. So this project is designed to reduce the discharge from the C11 West Basin and also maintain the flood control. So for this project, the final design was received in 2023, and the construction contract is planned to award it in the fiscal year 2024. For the water quality data, so this chart shows the C11 West Basin discharged from the S9 and S9A to the Everglades. So for the bar represents the TP load, and the dark blue diamond represents the TP fluid mean concentration for water year 2023. There are about 6 metric tons discharged to the Everglades, with the fluid mean TP concentration about 19 ppb. North Spring Improvement District is our last basin. This basin primarily with the urban and residential area and located in the Broward County. So for the NSID basin, and can discharge directly to the WCA2A through the NSID1 pump station, but most of the stormwater runoff are routed through the L36 north and south, routed to the Hillsborough and C14 canal, respectively, and so these, through these two canals, they discharge to the east. So the NSID sub-project is the Hillsborough Site 1 impoundment. So the long-term plan goal is to divert all the NSID runoff from the Everglades, so that depend on this sub-project. So this project, the phase 1 is completed, and the phase 2 needed the additional authorization from the Congress. Regarding the water quality data, so this again the same. The bar represents the TB load for each year, and the dark blue diamond represents the TP concentration. So for water year 2023, you see there are no discharge from the NSID basin to the Everglades. So from here also we can see there are minimum discharge after 2001, except 2018. That is also due to the tropical storm. The 2018 high TP concentration is resulted the default value because there are no sample collected during the storm. In summary, for water year 2023, the year basin TB load reduction is 63%, with the long-term TB load reduction 57%. And for the Sevan basin, is remained in compliance, and the measured TB load is below the target TB load. For the other tributary basin for water year 2023, voluntary BMP implementation and progress toward the completion of the third project. The continued success and ongoing improvement to the Southern Everglades Source Control Program, relying on the BMP implementation, research, and tracking program performance. More detail are provided in the Chapter 4 in the 2024 South Florida Environmental Report. And this is my contact information. Thank you.
Thank you, Dr. Wang. And we know that source control is critical, not just for those areas that are dispersing into the, directly into the Everglades, but certainly that input that we experience in the STAs. So thanks again. We're going to change gears just a little bit. We've heard a lot of data, STAs. We've heard a lot of information on updates to construction. This next talk is actually going to be how the public can access the system, and it's the public use on South Florida Water Management STAs. So Dan Carter, who is the section lead for Land Resources Bureau, is going to present this, and we look forward to hearing what he's going to share. There you go, Dan.
Is the red light on?
Yes.
Okay. All right. All right. Good morning, everyone. Dan Cotter, as Robert said, section leader in the Land Resources Bureau, and my group particularly oversees all the implementation of public use and management of recreation on district lands. Just by the way, the agenda had James Harbaugh, who works with me on there, but something came up, so he couldn't make it, so I'll go ahead and take. And what I'm particularly going to talk about, we have public use on all of our district lands, different types. I'm going to talk particularly about the public use that we implement on the STAs, and particularly the ECP STAs. So we get that question a lot, why have public use? And the main thing is, one is it's public lands, but also it's mandated, in Florida statute. And you can read that, some of that excerpt right there from the statute. But the important thing, particularly with the STAs, is that the public use that we implement is compatible public use. So, and it's probably our greatest or restrictive public use is on the stormwater treatment areas. What goes along with that is rules. We have the public use rule 47 you see there, and we go into rulemaking every so often, and that's what establishes what we're going to allow, how we're going to allow it. It also provides law enforcement, particularly FWC law enforcement, to enforce the rules that take place on district lands. And, the recreation we have on district lands and all district lands is, we always couch it as nature-based recreation. So we don't, have things like ball fields and that more active recreation. But the nature-based recreation on the STAs basically consists of hiking, wildlife viewing, particularly birding, which I'll talk about in a little bit, biking. We've got some picnic shelters out in certain areas, so there's time for picnicking. And in certain areas, usually outside of the cells, we have some fishing associated with that too. And then recreation within the cells is a very managed hunting in coordination with FWC. So partnerships, just going to touch real quickly on them and how important they are, especially with the stormwater treatment areas. Our responsibility is to open the lands up to the public, provide access, provide facilities and so forth, but we do rely heavily on partnerships, and just a few of them are listed here. I wanted to just go through those and give you some examples. FWC is a big partner with us on all our district lands, particularly for the management of hunting, so we work closely with them on those regulations and what we're going to implement as far as hunting goes. The Florida Trail Association, the Florida National Scenic Trail actually goes through quite a bit of district lands, and in particular for this presentation, STA 56, they actually have a route that goes right along there. They've got actually an area they can stop and rest and get water and a picnic shelter, so we work closely with them. Audubon Society, it's probably been 12 years or so now. We started, maybe even a little bit more, we started offering bird watching tours on the STAs. A few of the good ones, STA 56, STA 1 East, sometimes STA 2. Those are now sought after. I mean, people actually fly in from all over the country and even out of the world to go on those birding tours because it's actually an opportunity to drive the levees, highly managed, and see quite a few birds. They've created quite a few bird lists from those tours. And then we also work closely with a lot of clubs. United Waterfowlers is a big one that helps us manage some of the hunting. They offer some really neat opportunities, specialty hunts such as youth hunts, maybe wounded warrior hunts, so we work closely with them. We've got astronomy groups out there, believe it or not, some like STA 3-4, it's some of the darkest skies, and they take advantage of that. And then we've got many others, but these are a few good examples. Just talk a little, give you some examples of, and some of these I touched on, STA 56 there, you see the hikers up there in the corner here, up here. That's the levees that they access adjacent to STA 56 as part of the Florida National Scenic Trail. I mentioned the birding tours, how popular they are, especially in the season, which we're in right now. We've got a birding tour just about going out every weekend. STA 56, real popular. It also has an ADA boardwalk, which is used for disabled hunters, but it also works as a great birding platform. And then we have some unique things. Sometimes it doesn't happen, but once a year the flamingos may show up in STA2 and we'll run special, Audubon will work with us and run special birding trips out there too. And we produce maps similar to the one here that are on the website. We work closely with FWC and the partners such as Audubon on where, where on the levees they can go. STA 5.6 is quite a bit of access on the levees. And then our hours on the STAs, some areas, like our conservation areas, are open basically 24/7. The STAs are open just Friday, Saturday, Sunday, Monday during daylight hours. The weekends being the busiest, of course, and that allows then the midweek for some of the special veg management that may be going on, such as aerial spraying or something like that. Some of the other more specific recreation as far as the hunting goes, the waterfowl and alligator hunting is incredibly popular. The waterfowl hunting is probably some of the best in the state, if not the country. There's also plenty of alligators out there too. These are highly managed hunts into the STAs. It's very limited, very regulated, designated access points, what cells we're going to enter, which cells we're not, and I've got some examples of those too. And we offer some specialty hunts. I had mentioned that before, and you can see, youth hunt, I believe, down here. We've got wounded warrior hunts that are just great to see that are managed, where some of these people are just getting back into basically life, and this is one opportunity for them to go out and do something that they really enjoy. I mentioned fishing earlier. We do have some really good fishing areas. They're in general, if not all, are outside of the treatment cells. STA 1 East here, the northern portion there adjacent to our public parking lot, you're allowed to bank fish there. That's really popular. Those two cells, or those cells there, are not part of the treatment cells, so we're allowed to do that. STA 3-4, if you've ever been out to the Harold Campbell site, there's a boat ramp there. That goes into a water body that's outside of the, and to canals too, that's outside of the treatment area. Very popular, this area, and it's not on the slide, but A1FEB is also very popular. You can, there's individuals who can get, 50 to 100 bass in one day. A lot of this you've already heard, the science, the management, the construction. We, of course, got to work really closely internally with a lot of different departments to make sure we're, if we're having public access, particularly, for example, the hunting, where it's going, where it shouldn't go, that the main thing is we know there's a mission associated with the STAs. It has to do with water quality. So we want to always ensure that the public use doesn't impact that mission. And there's just some examples here. These are maps that we'll produce in coordination with FWC. This is STA 3-4, and it shows that this cell is off limits because it's going through a recovery of vegetation, and we don't want to impact that. Some of the other things we do too is we work close with our field operations. They, of course, got to go out during the week sometimes. Sometimes we might close an STA down on a Friday or a Monday because there's going to be an aerial spraying. So we work closely with them, make sure that they can still get their job done. Our land stewardship and our land resources, we have a biologist who is constantly going out and surveying a lot of different lands, but particularly the STAs. So whenever we see nesting, either on the levee or adjacent, and that could be snail kites even out in the marsh, we will close down certain levees to public use until that nesting has been complete. Of course, we work really close with our veg management team. You heard some of their presentations today. They do a lot of different things out there besides treating, the planting and so forth. And we want to make sure we're going and not impacting that, but through public use. And of course, construction. We've constantly having construction projects out there. STA 2 is a good example. We have several out there right now, and so we work around with them. And then this map here shows you, and this picture here shows you when we allow for the waterfowl hunting, we work closely with FWC. We always designate the parking lots so that the access is cross-flow and not with the flow. We do not want to cause short-circuiting out there. And this picture down here below shows you an example where the flow is going from left to right and the access is across that, so we do not impact or cause short-circuiting within the STAs. And along with alligator hunting, you know, I talked about construction and different things, but the one thing we try to do is we've got the public uses out there every weekend, and then we've got our hunting that takes place. We want to make sure that any activities that are occurring in the water are not impacting monitoring equipment or vegetation. And so, for instance, this is a map we work closely with FWC during the alligator hunts that along the southern portion of the STA, we do not impact the monitoring equipment, particularly the water quality it's leaving the STA. So we'll sign those areas along that levee. Veg management also is now implementing signage where they're doing, you know, sometimes spending thousands and thousands of dollars on plantings, getting the plants right. So they'll sign areas that we want to keep the public out of until those plants have established. I talked a little about construction. This is just one of many examples of STA 2 where they were doing the plugs. They had a lot of deadlines to meet. They were working on the weekends. So, and you can see by this map all the hashed areas where maybe construction was taking place or some other activities. So we may just limit, for example, hunting to 2 or 3 cells. And this year we had so much construction going on in STA 2 that the bridge that crosses from US 27 into STA 2 is usually open 7 days a week to allow people to get over to the northwest side of Water Conservation Area 2A. We're able to work with the contractors and the construction folks and just have a Saturday and Sunday access along this red line while the S620 construction was going on. So just some examples of how we have to work with a variety of different departments, but particularly with all this construction going on, we want to make sure that we're not impacting any construction. I missed a slide there. Okay. We have public meetings, and this is really important with our stakeholders, whether it's other agencies, but particularly the users out there and the public. We hold a rec forum meeting. The next one happens to be March 25th. That one's going to be on the West Coast in Collier County, and we're going to talk about the newly acquired green heart of the Everglades. But we have one in March, one in June and September. Those are very popular meetings for the public, very well attended. A lot of interaction. We talk about new projects coming up. We talk about existing recreation and improvements that we can make. And then we work closely with FWC on rules that they implement in our lands, particularly the hunting rules, and you can go on their website too and comment during their rule period. And this is generally how we work through, which we're doing a lot of these last several years with all the new projects and new STAs coming on and other areas too. We have recreation standards. Our engineers have recreation standards for building things such as boat ramps and boardwalks and parking lots and picnic shelters and all sorts of things. So when we have a new project come on, we'll lay out a plan. We'll present that at the rec forum, get input. Just at least show people that we're going to be providing public access. And then the project will go into construction, of course, particularly with the STAs and the reservoirs. There's a testing phase, and we take into a lot of considerations when those are being built as far as ADA considerations and what facilities and access points we're going to have. And a lot of consulting with the internal staff. We'll usually come back even to the public when we're getting ready to open it, talk about what types of activities we're going to have in there, you know, whether it's hunting, birding tours, what are the hours going to be, et cetera. Once that's done with the testing completion and projects, at some point then we go to the governing board, declare it, say an STA for the purposes of enforcing 47, and we open it up to the public. And that's it. There's my contact information if anyone has any questions or would like to reach out to me. Thank you.
Thank you, thank you. So, I want to thank all of the presenters. I think they did an excellent job, a lot of information. Many of you may have seen that we had comment cards that were at the table. And so we're going to move into a period where we have public comment. Those that are on Zoom, we ask that you raise your hand, and we'll acknowledge you. But we'll start with any comment cards that are in the room. I have one from Mr. Newton Cook. Mr. Cook, if you would, just use this podium to my immediate right, and, you know, share, you know, any information that you want to with our group.
Thank you, Newton Cook, United Waterfowlers of Florida. I didn't know the dam was going to be right before me, and I just really, we're so blessed at this water management district to have the people that allow public access from Orlando to the Keys. It's not just the SDAs, about 600,000 acres. And right now, turkey season's starting. And there are hundreds, at least, maybe thousands of people turkey hunting on district land this morning, or were for the youth hunt over the weekend. So I want to thank the district. I happen to be the fellow who facilitates the recreation meetings. I didn't realize we were getting a point on that. It will be March 25 in Maples, and well, it was June in this room. Come to that meeting if you like to recreate. Horseback riders, hikers, bikers, canoers. My favorites are stargazers. Mm-hmm. Come to those meetings and participate. And we outdraw the governing board members, I might add, the meetings. These today, I've been to this 20 years. Today I saw the greatest presentations on the SDAs. Dr. Tom James, he's still here, yeah. You don't know how much I've waited around and paddled around and fought to get these. SDAs were closed to everybody when they were first built. It took 5 years to get the first one open. 1 year, 15% of all the ducks in Florida were taken off 3 SDAs. That's incredible. Wow. Just incredible. This year, because we had a flock of fulvous ducks on 1 West, people came from all over the country to shoot just one. The SDAs are very important, but they are not managed for duck hunters. We understand that. But when they are managed correctly, and we have open water marshes with SAV, they are a magnet that literally attracts hundreds of thousands of waterfowl, not just ducks. I don't have to tell you that the mistake was made 6 or 7 years ago when the brouhaha about sending the water south, and we dumped a lot of lake water in the SDAs, which of course blew them out into cattails. We're spending millions of dollars fixing that up. Millions of dollars because we made the mistake of blowing lake water into the SDAs. There is a harsh political push to get more water in those SDAs from the lake. Don't do it. Last but not least, I pray I can get a group like this for Lake Okeechobee. The Lake of Okeechobee is being killed by the current management of the water levels. We need some scientists in the district helping us out. Thank you.
Thank you, Mr. Cook. Okay. I didn't need any other cards in the room, but I do have a few people that are on the line. Mr. Ben Olson. If you would, unmute your phone.
Hey, can you hear me okay?
Yes, we can hear you.
Okay. Thank you for letting me speak. Good morning. My name is Ben Olson. I'm a fourth-generation landowner in the West Feeder Canal Basin footprint. I'm speaking on the federal program work today. As mentioned earlier by Dr. Wang, South Florida Water Management District is the affiliated state agency. I'm limited on time, so I'll be blunt. This plan is foundationally incorrect. Without any evidence provided, the Corps and Water Management concluded that the Kissimmee Billy Strand, an isolated, pristine strand, was the native flowway. What the Corps' plan calls for is the backfilling of 2 privately owned and built canals to channel some 38,000 acre-feet of water through the headwaters of this pristine strand and then out through 72 square feet of culvert. The elevation data in this area was collected using LIDAR without ground surveying in conjunction. Then the elevation was averaged out into triangles, roughly the acreage of a section of land, and the acreage for each triangle varies. The variance in these triangles from the average height can be multiple feet, as the West Everglades is a series of ridges and sloughs. The modeling for this data shows there will be no major flooding. There's some issues with that, however. First, my math could be a bit bad, but I don't think you can flood 38,000 feet acres of water through 1,900 acres and then through 72 square feet without significant flooding. Second, the plan calls for a closable culvert system to be operated by the Seminole tribe unilaterally on the strand without any state or federal monitoring. The operation plan calls for the culvert on private land to be controlled by indigenous tribal ecological knowledge, and because of that, the tribe does not have to offer any explanation on when, why, or for how long the strand is severed. I have definitive proof this was never the native flowway. My great-grandfather built the road over and installed the culvert on the Kissimmee Billy Strand in 1963, 5 years before any of these canals built privately or by the Corps were operational. That culvert is still there to this day, and it's either 3 or 4 feet in diameter. If this was the native flowway, the roads would have washed away. The road isn't in their modeling. The modeling assumes the culvert to be open 100% of the time, despite being closable. This data is practically useless. What wasn't mentioned today is water management has backed off from backfilling these canals. The current board has correctly stated that this would be devastating for the wildlife corridor, but the federal plan must be stopped from authorization as it's designed currently to ensure the next board of water management doesn't implement this disastrous plan, which would wipe out 50,000 acres of primary and secondary panther habitat and sever a huge chunk of the Big Cypress Preserve from the rest of Florida. I filed sunshine requests with water management for communications pertaining to this project, but they slapped more than a $3,400 bill on that request. Their head of legal tells me it's declassified documents prior to allowing my constitutionally allowed inspection of public documents. He's refused to respond for 3 weeks now and also refuses to inform me specifically what statute water management is using to avoid inspection authorized by the Florida Constitution. We are without a doubt reaching the point of unreasonableness and bad faith on the part of the state agency. I've not sought legal representation yet, but it appears we're at a point where that might be unavoidable if Mr. Derek Brown cannot answer these simple questions to a member of the public. Real quick, for anyone listening that would like a comment of the Olson Ranch response or the one sheet with an alternative that would protect all of the wildlife corridor in this area while also achieving all stated goals, send me an email at olsonbenc@gmail.com. Once again, that's olsenbenc@gmail.com. All of this destruction is avoidable, but we cannot rush this bill through federal authorization. That only guarantees all of this beautiful land is killed at some point. Thank you.
Thank you, Mr. Olson. Thank you for your comments. I do not see any additional hands raised or any comment cards in the room. And so what I'd like to say is thank you all for coming to this meeting. We are, closing shop as the sun sets on restoration strategy in the next few years. And so we ask that you all stay tuned, and we'll try to keep you as informed as possible as things develop. So thank you all.